Bonjour tout le monde, c'est Alpha, cette conférence ULIS. Aujourd'hui, on a le plaisir de recevoir Kimberly Reynolds. Pour ceux qui sont là depuis quelques années, vous rappelez peut-être qu'elle avait donné une conférence en ligne en 2021. On avait prévu l'inviter en personne à ce moment-là, puis ça ne serait pas possible à cause de la pandémie. On est super content qu'elle puisse venir maintenant. Elle est là pour le congrès de Pathéo Laval, c'est un cousin de Pathéo Laval qui aura lieu le Pathéo Laval. Mais elle va donner une conférence différente. Elle avait assez de matériel pour donner les deux conférences, ce qui fait qu'on l'a invité à venir présenter aujourd'hui. Donc, Kim a fait ses études de baccalauréat en biochimie à Rice University au Texas, et ensuite elle a fait un doctorat en biophysique à l'Université de Berkeley en Californie jusqu'en 2006. Et ensuite, elle a fait deux postdocs, un en Californie au CRISPRi Research Institute, puis ensuite un deuxième où elle a commencé à travailler, c'est là que j'ai connu ses travaux, sur l'évolution des protéines à l'Université de UT Southwestern à Dallas. Et c'est là ensuite qu'elle a eu un poste de professeur, donc elle l'ont recruté à l'endroit où elle a fait son deuxième postdoc. Et puis là, maintenant, elle est fondable au Green Center for Systems Biology. Et elle est affiliée au département de bioinformatique et aussi au département de bioéthique de son institut. J'ai rencontré Kimberly la première fois en 2015, donc ça fait presque une dizaine d'années. On était à un événement à quelque part en Californie. C'est là que j'ai connu ses travaux. J'ai vu que c'est le genre de travaux en biologie du système qui nous intéressait et qu'on pouvait combiner avec l'évolution moléculaire. Et depuis ce temps-là, on suit ses travaux dans notre groupe. On a diffusé certains de ses papiers au club de lecture. Donc, on est vraiment super content qu'elle soit là. Et puis, c'est vraiment une personne qui occupe une niche unique dans le domaine de la biologie du système, à l'interface un peu entre la microbiologie, la modélisation, la biochimie fondamentale, la biochimie des enzymes. Et pour ça, elle a reçu de nombreuses reconnaissances, dont un prix en 2022 de Biophysical Society. Et aussi en 2020, elle a reçu un prix de son institution pour le mentorat exceptionnel qu'elle apporte aux étudiants gradués. Donc, c'est une personne qui est très impliquée en recherche et aussi dans la supervision des étudiants et de stagiaires postdoctoraux. Ces travaux ne sont pas sur des organismes modèles, mais sont financés aussi par le NIH, donc il y a toujours un intérêt pour la recherche fondamentale auprès des organismes subventionneurs qui subventionnent normalement la recherche en santé, aussi financé par le NSF, puis il y a eu des fonds aussi de fondations privées, comme la Gordon and Betty Moore Foundation, pour des travaux un peu flyés en biologie computationnelle puis en biophysique computationnelle. Donc, ce genre de fondation-là, ils aiment ça investir dans les jeunes chercheurs qui ont des idées novatrices qui sont très, très créateurs. Et donc, Kimberly a peut-être initié de certains de ces fonds. So, we're super happy that you're here. And,
thank you. Thank you, Christian, for the introduction and for hosting me. It's really a pleasure to be here and to have the opportunity to tell you all about some of the work in my lab. Okay, so more generally, right, I thought I would start off with kind of the big picture view of my lab. I've always been really interested in these questions of how genotype and protein ultimately relate to phenotype, right? And really trying to ask, how do changes in kind of biochemical or biophysical parameters at the level of individual molecules percolate through kind of metabolism and cell signaling and pathways to ultimately impact phenotypes at the organismal scale, right? And so my lab really broadly works on this question of, can we define kind of quantitative and predictive relationships that link protein activity, abundance, and sequence up to things like metabolism and phenotype, right? And we work on this problem most of the time in E. coli. And primarily, we work in E. coli because this is a place where we can generate really quantitative, high-throughput data that connects up these scales, right? So I can make thousands or tens of thousands or hundreds of thousands of perturbations at this scale by making sort of individual mutations in my protein or changes in gene expression, which I'll talk about today, and then measure in a very quantitative way the impact on things like cellular growth rate and fitness. And that means that I have a really large kind of data set that I can use to build quantitative models, whether they be more kind of mechanism agnostic machine learning models or more mechanistically driven models, kind of informed by kind of biochemistry and things like this. Okay. So for today, what I'm going to specifically focus on kind of understanding the connection between gene expression and, of course, downstream protein abundance and bacterial kind of growth properties, right? And much like in eukaryotic cells, where gene expression drives kind of phenotypic differences like differentiation, bacteria use variation in gene expression for all kinds of things, right? So they use changes in gene expression to be able to proliferate, of course, in different environments, different nutrient sources. This is this really cool data set that Jeff Barrick was involved in collecting, where they look at kind of changes in expression under hundreds of different media conditions. They can have genetic programs that allow them to differentiate into genetically identical but morphologically distinct forms in organisms like Colobacter, where they have these kind of swarmer and stalk morphologies, right? And then one phenomenon that I'm really interested in is that you can have these populations of cells that develop antibiotic tolerance, or what's called persistence, right? Where you have genetically identical cells and some subpopulation of them is not killed when exposed to high antibiotic, right? And then you can use these to reinitiate the population and grow. Which is just to say, in all of these cases, the changes are mediated not by coding sequence mutations, but by changes in the gene expression program. And so I was sitting with my students and we were thinking about, okay, we would love to be able to create some kind of model where we could predict how changes in gene expression and potentially environmental factors like nutrient availability or drug, so something like an antibiotic, how can we predict how these impact cell growth, right? And we envision this as almost like, we have little dials where I can turn up or down the expression of individual genes, or turn up or down the, kind of concentration of some factor in the environment. Can I, if I know the position of these dials, can I predict if my cells are going to proliferate really rapidly or if they're going to really not grow at all, right? And of course, if we could do this, that would be fantastic, because we could, in a more fundamental way, kind of understand evolutionary constraints on expression. I think a question many of you are interested in as well, right? Try to understand, like, how much of particular gene products do I need? Do practical tasks like engineering biosynthetic pathways to, say, optimize the yield or flux of some kind of bioindustrially useful product, right? And then understand the molecular basis of phenomena like antibiotic persistence. Okay. And so why is this problem hard and why haven't we figured it out yet, right? So I'll start with why it's hard, and then I'll tell you a little bit about what our proposed solution to this is. Okay, so I think here there's really kind of two key challenges in relating expression and protein abundance up to growth rate, right? And the first is that the mapping between kind of biochemical function and expression and growth rate phenotype is very nonlinear, right? And so here I'm showing this for just one enzyme that we've worked on. It's an enzyme called dihydrofolate reductase. Each one of these points is a particular point mutant that has variation in the biochemical activity. Now here we're changing directly the catalytic activity of the gene, but you could imagine that in some ways this is analogous to just sort of knocking down its expression, right? You're just reducing the activity of that gene in the cell. And what we see for this enzyme is that we can knock its activity down by, a couple of orders of magnitude relative to the wild type, and the cells still grow quite well. And then eventually they hit this cliff where they fall off and then they begin to grow relatively poorly, right? And so what this means is that, it's hard to build a predictive model because I can't just measure the two endpoints and tell you what happens in the middle. I actually need some fairly dense data collection to know what this relationship looks like. Now, the second challenge comes in the fact that these effects on growth rate are context dependent, right? And they depend on interactions between genes, or what we often call epistasis. And so the idea is now if I take the same set of about 10 different mutants, and I re-assay their effects on growth rate, but now in a different genetic background. So I've made a mutation just elsewhere in this biochemical pathway. You can see that now in this new genetic background, some of these mutations that had a pretty profound effect on growth rate before now have a fairly minimal effect on growth rate, right? And so we have this interaction that shapes this relationship. And these kinds of interactions, it would be nice to be able to sort of map them, know where they are, so we can build predictive models. Um, and kind of classically, these genetic interaction measurements or epistasis measurements have considered only binary perturbations, right? And so what I mean by binary is they can look at the interaction for the wild type gene and then a mutant or a knockout that's very strong loss of function. Okay, and so here I'm showing this as kind of this informal, like silly diagram of how we often look at interactions between genes, right? Which is in this kind of classic double knockout assay, which probably many of you are familiar with, right? So you start with your wild type reference genome. I make mutation 1, the cells are still totally fine. I make mutation 2, my cells are dead. And then I make the two mutations together, and now something radically different happens, and we would say, okay, there's epistasis because now we get this unexplained phenotype given the kind of phenotypes of the two single mutants. And then, of course, we can be more formal about this, and we can quantify epistasis in terms of a multiplicative model, which I'm laying out here, right? And so now I'm showing this in the form of a gene epistasis landscape. We have growth rate here on our vertical axis, right? And then we have gene knockdown, gene 1 knockdown here, gene 1 knockdown, gene 2 knockdown here, right? And so we can measure the effect of knocking out the two genes individually, knocking out the two genes together, and we can use this to compute epistasis, right? And so what this is it's the difference in growth rate between our double mutant here and then the multiplicative, so the product of the two gene effects individually. And so the idea is that if epistasis is equal to 0, we say these two genes don't interact. We can predict the effect of the double mutant from the single mutants individually. And if we get some non-zero value here, then we say, okay, there's some genetic interaction here that we can't explain simply that suggests these genes kind of functionally interact in some way. Okay. And so these kinds of epistasis experiments have proved, very useful, and they've long been used to kind of map biochemical pathways and study the organization of the cell. Here I'm showing an example from 2010 from Charlie Boone's lab, where people have done these kind of genome scale genetic interaction maps using lots and lots of double knockouts, and then using epistasis profiles to say, like, okay, here's a group of genes that show similar patterns of epistatic coupling, and so therefore they are involved in kind of similar biochemical processes. And so that's super useful from the perspective of annotating gene function, assigning pathway membership, right? But the challenge is that if you want to build a predictive model that now relates gene activity or gene expression to growth rate, we don't know what happens in these kind of regions of incomplete knockout or incomplete knockdown, right? And so what we'd like to be able to do is to understand what is happening kind of more completely along this surface that relates gene knockdown to growth rate. And so the parallel I'd like to draw is this is sort of a digital perturbation, right? Where you have 1, 0, it's on or it's off. But we'd like to be able to move to a more kind of analog measurement, right? Where now we can have different levels of gene expression and kind of measure out the effect on growth rate, right? And then, here we have this multiplicative measure of epistasis, but what would the analogous measure of epistasis look like for something more like this? Okay. And it's just to say that, of course, if we do this sort of binary perturbations and we only consider the endpoints, then we're often going to miss kind of interactions that occur at only intermediate strengths of sort of knockdown or biochemical activity, right? Because we only see these two endpoints. And that's especially true amongst essential genes, where if you knock out one of the genes, the cells are just dead. And so you can't really see kind of the broader structure of interactions. All right. So this is what we set out to do. We wanted to define a new kind of continuous or analog measure of epistasis that would allow us to kind of capture the nonlinear relationships between gene expression and growth rate. We want to be able to summarize information across perturbation strengths, right, and maybe different environments. So how can we measure genetic interactions across this kind of range of knockdown effects? Ultimately, we want to only have to make a limited number of measurements per gene pair because we appreciate that we do not have infinite experimental capacity, right? And then finally, we want to be able to predictively model. So we want to be able to go beyond the experimental data we've collected to predict things that we haven't yet experimentally measured. Okay, so these are the folks who took on this project. Andrew Mathis is a graduate student in the lab who really developed the kind of experimental component of it, the CRISPRi tool, so I'll tell you about in a moment. Ryan Otto and Agata Turska-Novak really drove forward the modeling component of it, this continuous epistasis model that I'll tell you more about. And then Phil Brown is the newest student in the lab who's now pushing this work forward to explore interactions and whole pathways. Okay, so we want to build this model, and we really kind of need two things, right? The first component that we need is we need a high throughput experiment where we can essentially, change the strength of these dials. We want some way to be able to go into the cell, titrate the expression of individual genes alone and in combination. We want to be able to titrate environmental factors and measure out growth rate in really high throughput. And then the second component we need is a computational strategy to summarize all of these data and begin to make new predictions. Okay, so first I'll show you the experimental part, and then we'll talk about how the computational model works. So the experiment that we're doing is with a tool called titratable CRISPR interference. And first I'll just start with introducing how CRISPR interference works in bacteria. Okay, it's a little bit different than how it works in eukaryotic cell systems. But basically the idea is that we have a guide RNA, that's this little kind of black and tan piece here, and it assembles in a complex with dCas9, which is a catalytically inactivated dCas9 endonuclease. So it no longer cuts DNA, right? But the guide RNA directs it to the genome, and then it just parks itself there and becomes a roadblock for RNA polymerase. So what this does is it generates kind of a block of transcription, and it generates a strong knockdown, but not a full knockout. And so it's inducible. We can turn on the expression of dCas9 and take out the expression of a targeted gene. Okay. And now we can combine this with what's called titrating guide RNAs. And I'll spend a little bit of time to discuss these further and how we design these. But in essence, the idea is that we go through and we introduce mismatches or mutations in these guide RNAs. There's a 20 base pair homology region that tells the guide RNA where to stick. And by introducing mismatches into this 20 base pair region, we can make it so that it has reduced affinity for the DNA, and we have less repression strength. And so basically, the more mismatches you have, the less strongly you repress transcription. And so we can combine these two things, these titrating guides and the CRISPRi, to get, excuse me, a variety of what we're going to call repression efficiencies. So this is measured with RT-PCR or qPCR. A repression efficiency of 1 is near complete knockdown, right? So the gene is maybe 95, 98% knocked down. A repression efficiency of 0 means wild type-like expression, right? So these are very highly repressed, and then as we introduce mismatches into our guide RNAs, we can kind of step down the repression efficiency back to wild type. Okay. And now the convenient thing about this is that the guide RNA itself can serve as a DNA barcode in high throughput sequencing-based experiments. And so what we do is we build a really large guide RNA library that now has, 10 to 12 guide RNAs targeting multiple genes. We take the library and we transform it into a strain that harbors dCas9. And so we have our strain here. Each of these colors is supposed to be a different variant. We grow them under some selective conditions, and we see their fraction changes. And then we use next generation sequencing to monitor the frequency of each guide RNA in the population over time, right? So for those of you who do deep mutational scanning, it's entirely the same concept, except now instead of sequencing mutants, we're sequencing guide RNA sequences. And then we count basically the number of each guide RNA variant in our population relative to a non-targeting guide that has very minimal effects on growth. And we look at that frequency relative to T0. So something like this green variant here, we can see is dropping out in our population over time. And we can fit a line to this, and that gives us a selection coefficient or a relative growth rate that describes the kind of fitness of this particular knockdown. And then finally, now because we have multiple guide RNAs targeted against a particular gene, we can construct these kind of growth rate versus repression efficiency maps and use this to measure growth rate effects to sort of thousands of variants in a single experiment. So that's the overview of the experiment, and then I'll share with you a couple of technical details that I think are important to doing these experiments really well. Okay, so one kind of, I think, important design feature of these, like why do we use these titrating guide RNAs? Others in the literature have used kind of varied dCas9 expression. So another option is to have dCas9 under an inducible promoter and just turn down or turn up its expression to titrate your knockdowns. David Picard's lab has actually now shown that if you titrate Cas9 induction, you can indeed titrate the knockdown level, but that you have these kind of messy subpopulations of cells, right? So here the reporter for Cas9 activity is GFP expression. And you can see that at various inducer concentrations, you actually get a pretty broad distribution of knockdown effects. Whereas if you use these titrating guide RNAs, you get these nicer kind of tighter distributions of knockdown effects, right? So we really like these. And then the other possible thing is that you can take all of these and grow them in a single culture. You don't have to compete at sort of different inducer concentrations. And this allows you to do the experiment in a more straightforward and high throughput and high resolution way. Okay, and then another kind of important aspect of this is we use internal barcodes on our guide RNAs to estimate experimental noise. So for every guide RNA in our library, we construct our library 6 times, so they have 6 kind of independent barcodes. And then we can look for a particular experiment at the correlation between, say, barcode 1 and barcode 2 to estimate the noise in our experiments. And then we also use these for something that we call escaper correction. So one of the known phenomena in CRISPRi is it is not particularly stable in bacteria, especially when you're targeting essential genes. There's enormous selective pressure to sort of shut down the CRISPRi process, either through inactivating the Cas9, mutating the genome itself so that the guide RNA doesn't stick, right? And so we can identify these escapers by looking at what the behavior of our 6 different barcodes. So this is a set of guide RNAs targeting a gene called NERD-A. And you can see 5 of our barcodes are doing one thing, and the 6th barcode is doing something different. So presumably it has escaped CRISPRi targeting. And so we can go through our data and kind of statistically identify and correct for these cases. And what we found is that when you apply this kind of escaper correction, you consistently get, and it makes sense, sort of lower estimates of selection coefficients and fitness than if you don't apply this correction. Okay. And then finally, the last part of this is how do you actually design guide RNAs to make these titrations? There's been multiple kind of proposals about how one could approach this, right? So if I have a 20 base pair homology region, it's going to stick to my gene, right, to induce my knockdown. And it has at the end of it this thing called a PAM sequence, which is absolutely necessary for Cas9 recognition, right? And so there's a couple of ways to approach this. One is you could make sort of single mismatches or single mutations throughout the homology region. You could make sets of double mutants, or you could make what's called compounding mutations, where you basically walk from the region that is distal to the PAM, and you systematically go through and introduce a mismatch, right? So just base by base, you flip these until you introduce more and more mismatches. And so we said, okay, we want to try these different guide RNA design strategies to figure out what is the best way to design these so that I can get really kind of systematic, gradated, titrated steps in gene expression. All right, and so to do this, we said, okay, we're going to create guide RNA libraries, and we're going to do this across 88 different growth-linked genes in E. coli to see how they behave. We chose these genes to sample a variety of cell processes to be positioned kind of across the E. coli chromosome. They're all growth linked, 76 of them are essential genes, and they span a pretty wide range of protein copies per cell. So these are 88 genes. And then we can look at the results. Okay, so this is a heat map of our full library. So to walk you through it, we have normalized growth rate is indicated in this kind of purple to yellow color map. Wild type like growths are around 1, this yellow color here. And black to purple means that it's almost dead. On the horizontal axis, we have our set of 88 genes. We have a handful that seem to have been missing from our library or dropped out very early, so we couldn't actually compute growth rates. And then we have our different guide RNA design strategies here on this vertical axis. So we have single and double mismatches here, and then we have these compounding mismatches here. And I think what you can probably already start to see just from this graph is that visually, by introducing these compounding mismatches, we can start to get these kind of systematic titrations from things that grow fairly poorly to things that now grow pretty well. And to show this in sort of a different way, right here again, we have our different guide RNA strategies. And you can see that as you sort of systematically add in mutations to the guide RNA, you can have a kind of titrated effect on growth rate and presumably the underlying gene expression, right? And so going forward, this is our strategy for designing our guide RNA libraries. What we do is to basically design a series of guide RNAs where we target 2 positions in the gene, one very near the 5' end of the gene, one a little bit further in that has a less extreme effect. And then we introduce sort of either no mismatches or that we sample the number of mismatches that get kind of these intermediate effects on growth and expression. And so through this empirical design strategy, we can create guide RNA libraries that have sort of 5 to 6 resolvable growth rate effects and presumably sort of similar numbers of effects on gene expression under the hood. Okay. So that's one thing that I'll say is an open problem in my lab. Right now, when we design our guide RNA libraries, we have this kind of empirical approach for designing and selecting them based on kind of numbers of mismatches that we know do about what we would like, right? But one thing that we're very interested to do and are working on now are kind of designing quantitative algorithms and predictive models to be able to do this in a very kind of prescriptive way for individual genes and know for a given guide RNA, can we predict the transcriptional effect it's going to have? So that's something that's in progress now. Okay. And this is just, again, to make the point that now if you have the strategy where you can do these kind of single gene titrations of expression, is you can start to pull out interactions that you would miss if you just did this at the limit of wild type and a total knockout or wild type and a very strong knockdown, okay? And so here we can see this for this gene called whole B. Here I'm showing data for 2 genes. These are 2 of our 88 genes that we targeted. And we assayed the entire library in 2 environmental conditions. We looked at the growth rate effects in glucose and then also in glycerol. And so for genes like PFKA, this is almost a control. It's a gene that's involved in glycolysis. And so we see in glucose conditions that we get this really kind of sharp growth rate defect. And basically there's a threshold for which if we have a sort of very few mismatches and a really strong repression efficiency, these cells are dead. And then this goes away if we repeat the experiment in glycerol where this gene is no longer essential. But then we can also identify genes like whole B that's involved in DNA replication, to our knowledge is not involved in glycolysis, and yet we see that kind of intermediate levels of titration, we get these growth rate differences. And so in fact, in our library, we can identify kind of a set of 14 different genes that show an interaction with the environment that we would miss if we were only considering kind of endpoint measurements of the system. Okay. So that's our general experimental approach. And now we want to take this forward and combine it with some modeling to be able to sort of build predictive models for cell systems, right? Oh, okay, wait, so I should tell you also, right? So these are all now for single gene effects, right? So here I'm titrating just, 1 gene at a time, and I'm doing this in 2 different environments. But the cool thing is now we can start stacking these up inside of a plasmid, right? So instead of just titrating the abundance of 1 gene, I can now have guide RNAs targeting a whole other set of genes, right? And I can, of course, make these in all possible combinations. So I can make lots of different pairwise knockdowns, and I can begin to actually construct these pairwise gene knockdown landscapes where I titrate the abundance of gene 1, titrate the abundance of gene 2, and measure out the growth rate effects, right? I'll show you data today a little bit later on where we've done this now for 3 guide RNA sets. We've gotten up to 4. I know someone who's made up to 7, right? And I think there's a question of like how many combinations and how far can you formally push this? But I think it's very exciting to be able to sort of explore these changes in very high dimension. Okay. So we want to use these data and try to combine them with some predictive modeling. And to develop our approach, we said, okay, we're going to start with 9 enzymes where we have a pretty good idea of what they do and have sort of different pathway architectures, right? So we can ask that, does our model kind of capture genetic interactions and relationships for different kind of biochemical pathways and interaction types? And so we chose these 9 enzymes. There's 2 that we chose from dapaminoleic acid synthesis that's in front of lysine metabolism, DAP-A and DAP-B. We picked purine and purell and purine metabolism. So these are both examples of kind of serial catalytic reactions. We picked 3 genes that are involved in a cycle in folate metabolism. And then we picked a pair of genes that catalyze parallel reactions, GDHA and GLP-B. And so these are interesting because they form what's known as a synthetic lethal. If you have one gene, the cells will grow, but if you knock out both, then the cells are dead, right? Because they have this kind of redundant relationship. Okay, so we have these 9 enzymes. We're going to design a guide RNA library that targets all 9 of these and can target them to different abundances so we can titrate their expression. And this is what the growth rate data look like, right? Okay, so I'll take some time to walk you through this. Again, we're showing the color indicates growth rate. 1 or white is wild type like growth, and then this dark blue color is something that's near lethal. This first column is all of our guide RNAs paired with a non-targeting guide RNA. So these are basically the single gene effects. And then each one of these squares are pairwise interactions between guide RNAs targeting 2 genes. So for example, purell and DAP-A. And the guide RNAs themselves are ranged from kind of a minimal repression efficiency to something with very strong repression efficiency, right? That has almost no mismatches in the guide RNA. And so you can see that, for example, like for this purell and DAP-A pair, that as you repress purell, the cells get more and more sick. They grow less and less well. And then similarly for DAP-A, you have a more modest growth rate effect, right? And then in these kind of pixels in the corner, now we're beginning to examine the interaction between these 2 genes. Okay, and so you can see these kind of like dose-dependent growth rate effects throughout our set of 9 metabolic enzymes. And we said, okay, now we can go through and we can use these to calculate epistasis or genetic interactions in kind of the usual way, right? And so what I'm doing is I'm calculating now the epistasis between each pair of perturbations using the standard multiplicative model. So I have the growth rate effect of the 2 guide RNAs together minus their multiplicative effect of the 2 guide RNAs on their own, right? And so in the limit that these don't interact, I should see sort of 0 epistasis. You'll notice that for much of this upper diagonal part of the matrix, that's true. There's sort of not that much interaction. Kind of makes sense because these are drawn from a variety of different pathways. And then we see a subset of things where we get these strong positive interactions. These are buffering interactions, right, where the effect of the 2 genes together is less extreme than you would predict from the 2 individual components. And then we also have these negative interactions, which correspond to a more extreme effect than you would expect from the 2 individual genes alone. And you can start to see some things that make sense here, right? So we see positive epistasis for an enzyme pair that does serial reactions. So for DAP-A and DAP-B, we see this kind of positive or buffering epistasis. And that kind of makes sense. Like if I knock down DAP-B in the background of DAP-A, it's not going to get worse because it's already impaired for lysine metabolism, right? And so we see this sort of positive epistasis. And then similarly, for this pair of enzymes, GLP-B and GDHA, that are synthetic lethal, we get this really strong negative epistasis because as I titrate one, nothing happens. As I titrate the other, nothing happens. And then in this bottom part, the cells are quite sick because now I'm knocking down both of them, right? Okay, so how well is data? I have all these epistasis measurements, and the question is, how do I use this to actually understand these pathways, right? I certainly don't want to go out and publish a paper that now has, thousands of epistasis measurements. It seems a little bit nonsensical. And so we want some way to kind of summarize these data more succinctly and go beyond mapping to being able to predict kind of these growth rate effects, right? For perturbations that are, outside the set that we've made here. And so to do this, we drew inspiration from a body of literature that works on kind of predicting the effects of antibiotic combinations. In particular, our model is inspired by this work from Anat Zimmer and Uri Alon's lab. Sorry. And their question was, okay, if I'm administering different combinations of antibiotics or potentially different combinations of chemotherapeutics, can I make a limited number of predictions and then, limited number of experimental measurements and then predict the effects of these drugs in combination, right? And the idea here is to really be able to design kind of combination therapies for antibiotics or chemotherapeutics. And so we were reading this paper and we got really excited about it because we said, okay, this is really an analogous problem here. Because in their case, they're titrating the dose of two particular drugs, right? Measuring the effect on growth rate, and then trying to predict the effect. And in our case, instead of now titrating drugs, we're titrating pairs of genes, and then we want to measure how that affects growth rate. So we said maybe we can sort of borrow their framework and apply it to our data. Okay, so that's what we've done. So first of all, we're going to measure each of our single gene effects as a sigmoidal kind of dose response curve. And so for those of you who have worked with antibiotics, this is basically an IC50 curve, right? So we're fitting this sigmoidal function. We have this parameter called R naught, which is analogous to an IC50. It's the repression level at which our growth rate goes to a half, okay? And then we have N, which is a Hill coefficient, which tells us how steep this relationship is. And so now we can ask, does this fit kind of our 9 enzymes, right? And so we can fit these 9 dose response relationships to our set of enzymes here. And in general, we find that it fits reasonably well, with the exception of maybe GDHA and GLIT-B. Because these two perturbations are not deleterious on their own, we can only sort of very poorly constrain our R naught and our N. But I'll show you as we move forward that because we're going to measure these in different genetic backgrounds, ultimately this ends up being okay. Okay. And now the kind of interesting part comes in, which is how do you capture kind of pairwise interactions now between genes, right? And so to capture this, we introduce this idea of an effective repression level. And so the concept here is if this is the dose response curve for our gene in blue in the wild type background, let's say we now remeasure it in a different genetic background and we get this orange curve here. Well, now what we see is that the growth rate is behaving as if this has an effective repression level that's kind of more extreme than what it actually has, right? So we have sort of a different effective repression level in this new genetic background, right? And that effective repression level could be sort of more severe, or it could be buffered, right? It could go in this direction. And so we capture this effective repression level with this new measure called RAF. So this is our effective repression level. It's equal to the actual repression level that we've measured. And then we divide it through by this factor that now depends on the interaction between pairwise interaction with all other kind of genes in our system. So we enumerate over all other J, so these are all the other genes in our system. And then we have this coupling constant that says how strongly does gene J modify the effective repression level of gene I, right? And so this coupling constant, if it's negative, we have the effect of kind of decreasing R naught. If it's positive, we increase R naught. The direction is not super important, but the main thing is that, you can see if this goes to 0, the second part of the equation goes to 0, RA effective is equal to RI, and there's no coupling there at all. Okay, so we have this pairwise model, and we can ask how well does it describe our data. And so to establish a baseline here, we're just kind of consider this for a particular pair of genes, DAP-A and PURIN. And basically this first column here corresponds to one of these sigmoidal titrations for DAP-A. This row here corresponds to the titration for PURIN. So we can fit these two relationships, and in the absence of any coupling between these genes, we would predict that this would be the pattern of pairwise knockdown effects, right? Given the data in this first row and this first column here. So this is our prediction with no couplings, and we can subtract this matrix from this matrix to compute an RMSD and say how much does our model deviate from the data. And you can see that, we kind of have this region in the bottom right where we predict that growth rates are sort of worse than they are. So now we can repeat this by adding in coupling here. So we add in our coupling constant. So we fit this AIJ term using these pairwise measurements. And you see that now we can get much better agreement between our model and our experimental data, and it's reflected in a lower RMSD, right? And so for those of you who are familiar with model fitting, you're going to say, okay, that's great, but of course the fit's better. You added kind of 2 additional parameters in terms of the couplings between DAP-A and PURIN, so of course it fits better. You can show with an Akaki information criteria analysis that it's statistically justified to include these extra parameters because of the improvement in the fit. Okay, and so now we can do this now not just for DAP-A and PURIN, the pair I showed you earlier, but for all pairs in our data set. And you can see that pretty systematically that our epistatic model that allows for these pairwise couplings has a lower RMSD to the data than these null models here. You can see one of the most extreme cases is for GLIT-B GDHA, the synthetic lethal pair, where the null model completely by definition misses the interaction, but we can now include these coupling constants that allows us to start capturing the fact that they're synthetically lethal. And so by and large, our performance is better. There's a subset of genes on the diagonal that presumably have pretty minimal epistasis, and it works well either way. Okay, and now these coupling constants, these AIJs, give us kind of a summarized description of epistasis. So now instead of reporting all of these measurements, I can tell you, okay, these are these kind of coupling coefficients between pairs of genes. Interestingly, they're asymmetric because I have a coupling between GLIT-B and GDHA, and then the other direction between GDHA and GLIT-B. In some cases, this asymmetry seems to have some meaning. In other cases, not so much. That's something we're trying to understand a little bit better. All right. And then finally, the pattern of couplings that we infer through this approach, we see is much more sparse than when we consider just sort of binary perturbations. So if we calculate the couplings between these pairs of genes, just considering our maximum knockdown effect, right? We can compute sort of like a more classic genetic interaction or epistasis score. We see that our model, the kind of classic epistasis would say there's lots of interactions. And in fact, we kind of smooth these out when we consider all of these intermediate levels, right? So I think a lot of these are coming from the fact that considering only the limit of total gene knockdown gives rise to a lot of noise. Okay. So now, all right, so I just told you this works. We can describe it. We can fit these models. But this is a lot of measurements, right? This is like almost 100 measurements for like one gene pair. If we want to scale this to whole pathways or entire genomes, this is going to get kind of messy pretty quickly. So we wanted to ask, like, do we actually need all of our data, right? Or could I throw away a substantial portion of it and still build a model that now predicts all the measurements that we've thrown out? And now we find actually the model is quite robust to subsampling our experimental data. And so what I'm showing here is we basically did 100 iterations of kind of randomly keeping a subset of our measurements, fitting the model, and looking at the RMSD to our data. And you can see that you can throw out about 80% of our measurements and still get a model that has an RMSD that's pretty close to the complete data set, right? Of course, in the limit that you throw out many of the measurements, then you start to get close to this null model RMSD. And so what that tells us is we can now build sort of more minimal libraries to compute these coupling constants and then be able to predict kind of interactions. Okay, so what else can this model do, right? We have a way to describe pairwise interactions, but the real power of this is if I could kind of collect data for pairwise perturbations and now begin to predict the effects of sort of 3 or 4 or more perturbations at a time. And so that's what we tried to test here. So the idea was, okay, now instead of looking at kind of pairwise libraries, can I instead construct libraries of sort of 3 knockdowns at a time? And so we made a library of sort of about 1 million constructs. So these are combinations of 3 different knockdowns. Actually, this is really strongly bottlenecked, right? So here in our final library, we end up with about 830 constructs that target sort of 3 genes at a time. We have a smaller subset that target only pairs, and then we have 4 single knockdowns. And we say, okay, we have these data. Can we predict these data using only our pairwise measurements that we made before, right? So can we predict these 3 gene effects from measurements on 2 genes at a time? And the answer is yes, and it seems to pretty well outperform the null model. So these are the growth rates you would predict with a model that does not include any coupling. This is the predictions that you get with a continuous epistasis model. And you'll see that it kind of corrects for 2 errors. One is this kind of systematic shift away from the y equals x axis, where here we sort of systematically predict that the growth rates are worse than they actually are. And so this gets kind of corrected globally with the continuous epistasis model. And then the second thing is for pairs of genes where we know that there is epistasis, that we can now sort of bring these back in and predict these more carefully or with higher accuracy. Okay. So we can predict things for sort of 3 gene perturbations at a time. And we said, okay, well, what about like a 4th order interaction, okay? And, you know, technically our modeling framework should not be limited to sort of titrating individual genes, but we could also start now to titrate an environment. So here what we've done is we're going to target two different enzymes inside of folate metabolism, this gene FolA and ThiA. So we're going to titrate their abundance. And then we're going to titrate the abundance of these two different environmental factors, methionine and thymidine. And we chose these because they're products of the folate metabolism pathway. And so we expect there to be, a lot of kind of interactions or epistasis here, right? And so to consider kind of a scale of possible combinations here, if we think about, so we made sort of 10 knockdown levels of gene 1, 11 knockdown levels of gene 2. We can have, 6 or 7 knockdown levels in the environment. So in total, this would be like 34 single measurements, not so bad. 561 pairwise measurements, that starts to get a little bit more, right? But in total, there'd be 4,620 total combinations if we were to consider kind of all possible combinations of these. Okay, so, first we started off and we said, all right, we're going to collect kind of a training and test set. And we're not going to make all of the measurements, but we're going to make a little under half of them. And so what the data look like is each one of these squares is now titrating the abundance of FolA by ThiA, right? So those are titrating the two genes in one square. Both of these are essential genes. So you can see as we titrate down activity, the cells die in both directions, right? And then we can repeat that experiment now in all of our different environments. So in this way, I'm increasing the concentration of methionine in my media, and in this direction, I'm increasing the concentration of thymidine in my media. And so you can see some things that start to make sense. There's some pretty strong environmental dependence. And so, for example, as we titrate the gene ThiA, we should get that the cells die. But once we hit some critical threshold of thymidine, we no longer see this loss of function with ThiA titration because you're supplying an exogenous source of thymidine that's now rescuing growth, right? So we see that kind of in this direction. And then we also see some buffering of growth rate effects in the methionine direction, right? Okay, so we have this full set of measurements. The gray ones, of course, ones we didn't measure. And we want to ask, how well can we predict these? And so first you just start with kind of the simplest thing, right? You say, okay, what happens if we only measured the single gene effects? I'm not going to measure any pairwise interactions, but I just have 34 measurements, and I can fit the single gene effect curves, and I can also fit single environment effect curves, right? So I fit these sigmoids that now describe the relationship between growth rate and repression of FolA, repression of ThiA, so my two genes, and then changing the environmental conditions, the amount of thymidine or the amount of methionine in the media, right? So I fit these 4 curves and I say okay, now I can sort of plug in different repression levels and different combinations of these, multiply them all together, and try to predict what happens for different combinations. How well do I do with just these single measurements? And the answer is terrible, right? So if I only use these single gene effects, the predictions are awful, right? So if I have predictions versus measurements, you can see that there's really kind of no relationship here, and the RMSD is pretty high. So clearly we need more data than 34. So now we can move up in order of magnitude. And we can say okay, we're just going to use our subset of pairwise measurements we have. So again, we're going to use our 34 single gene effects and single environment effects to fit the sigmoids for each of these. And then we're going to use our pairwise measurements to now infer coupling. So we're going to coupling between FolA and ThiA, FolA and thymidine, FolA and methionine, right? We're going to get all the pairwise couplings. And then we're asked, okay, given this, how well can we predict our data set? Now, 425 measurements is still a lot, but it's an order of magnitude less than the 4,000 kind of possible measurements for the system, right? And the answer is that you do much better. Is it perfect? No. We'll talk about that in a minute. But you now get reasonable correspondence between your growth rate predictions and your growth rate measurements for this much more sparse data set. And so what this tells me is that you can get a pretty vast improvement in model fit with kind of a small increase in training data if you just allow yourself to fit these pairwise couplings and you neglect all possible higher order interactions. Okay, so then you're going to be asking what are these orange points over here that we don't really do that well on? So we'll zoom in on these for a moment. These points here are actually from a true third order interaction between our two genes, FolA and ThiA, and the environmental condition of thymidine. And so here we have our measured growth rate effects. Here's how the null model does, which is not very well. This is what it predicts. And this is what our continuous epistasis model predicts. So this is FolA and ThiA knockdowns in the absence of thymidine, and this is in the presence of thymidine. So in the presence of thymidine, our model gets correct that Thi knockdown no longer has an effect, right? It can account for the coupling between Thi and thymidine in the environment, and that's good. And the part that we miss is actually hard to see here. These pixels are actually slightly pink, right? And so what we find is in our actual experimental measurements, these have sort of a growth rate rescue effect, whereas if you knock down these two genes together, you actually get pretty good growth rates, and we miss this in our continuous epistasis model. So there's a real third order interaction here that we miss. And so that's one question I'm interested in going in forward is like, are these kinds of third order interactions between genes and environments common, or are they kind of rare phenomena? And we can identify them by looking at deviations between our model and experimental data. So that's basically the idea, is that now we have this approach to kind of consider genetic interactions genome-wide. This is something we're kind of scaling up to try to cover a good chunk of the essentialome in E. coli, where we want to make sort of dense single perturbation response curves describing the dependence of growth rate on individual genes. And then we're going to sparsely collect these pairwise measurements, use them to fit couplings, and then now be able to predict kind of additional pairwise and higher order phenotypes or kind of combinations of knockdowns or combinations of knockdowns in environments. And then I have some more slides about future directions, but I think I'll end there to give some time for questions and just thank my lab. They're a really fantastic group of people to work with, and I welcome any questions.